This is the PowerPoint lecture for Chapter 26, and we're also going to touch a little bit on Chapter 25. So this first slide has a lot of definitions. Evolutionary biology is about two things. It's about the process, which includes natural selection and the origin of species, and also history. And a major goal of evolutionary biology is to reconstruct the history of life on Earth. Systematics is the study of the biological diversity in an evolutionary context. And part of the scope of systematics is the development of phylogeny, which again is the evolutionary history of a species or of a group of related species. Some related concepts we'll touch on are taxonomy, which is another way of classifying organisms. And then the definition for systematics is the studying of biodiversity in an evolutionary context, particularly using the fossil record. And then the derivation of the word phylogeny comes from phylon, which is tribe, and geny, which is genesis. So how we come about with the beginning or look at the progression of a particular species and genus, the tribe, over time. So some of the important points of Earth's history, and these of course are estimates, but these are some things for us to focus on. And as you look at Chapter 25, there are just a couple sections that you need to look at, but these are some of the major concepts that you should review. The Earth is about 4.6 billion years old. Life on Earth emerged 3.8 to 3.9 billion years ago, and that, of course, was very simple prokaryotic life. The Oparin-Haldane hypothesis, take a look at that. Miller and Urey's experiment. Look into and study Earth's early atmosphere, what it was believed to consist of. And also, you should know that RNA was the first genetic material. At least that is the belief. It's called the RNA theory. And also, we've already talked about this earlier, the endosymbiotic hypothesis, which states that there was a mutualistic fusion that occurred where mitochondria and chloroplasts were made part of another cell that later developed into our eukaryotic cells. So fossils, they are our only clues to life in the past. They help us to learn about evolution by acting as a type of photograph of an animal at a particular point in the history of Earth. So if you see here matching these up, you can come to guess by working with fossils what the morphologic characteristics of an organism were. Fossils also help us understand what kind of environment existed during the time the fossil was made. For example, by studying fossils, a paleontologist can learn that 440 million years ago, Ontario was covered by a warm, shallow tropical sea that had corals, trilobites, sea lilies, clams, and other creatures. And it's hard to imagine something so landlocked at this point was once underwater. Some ancient animals like trilobites have no close living relatives, while others do have close relatives living today. So by studying living animals and how they live, scientists can better understand how animals lived in the past. Just to review how fossils are formed, sedimentary rocks form layers of sand and silt that settle to the bottom of seas and swamps. And as these deposits pile up, they compress the older sediments below them into more and more solid rock. And while this is occurring, the bodies of dead organisms settle along with the sediments and actually get pressed into this layering that's occurring. But only a tiny fraction of these dead organisms do get preserved as fossils. We have what are called index fossils, which are keyed to the relative time scale. They're forms of life which existed during limited periods of geologic time, and they're used for this reason as guides to the age of the rocks in which they're preserved. So they're essentially key fossils that we know about the time period where they existed as living organisms. So as new fossils are found, they can be compared to these to get relative dating. So in terms of relative dating, the geologic timescale consists of eras and periods and epochs. And so we date a fossil based on where it is found in the rock strata, and that is our relative dating. Newer eras are marked by a distinct change in the type of fossils found. So by comparing different sites, geologists have established this geologic time scale with a consistent sequence of historical periods. And these periods are grouped into 4 eras: the Precambrian, the Paleozoic, the Mesozoic, and the Cenozoic eras. Boundaries between these eras and periods correspond to times of a lot of change going on, particularly when there were mass extinctions. And the serial record of fossils and rocks helps to provide relative ages of the different fossils that are found. But they're not the absolute ages, which would be the actual time that the organism died. Absolute dating of fossils is usually done by radiometric dating because fossils contain isotopes of elements that they accumulated when they were alive. This technique takes advantage of that fact, and the isotopes undergo radioactive decay in which that isotope of the element is transformed to another element. For example, the radioactive isotope of carbon-14 is present in living organisms in the same proportion as it occurs in the atmosphere. But when the organism dies, the proportion of carbon-14 in relation to the total carbon declines or goes down as the carbon-14 decays to nitrogen-14. So an isotope's half-life is the time it takes for 50% of the original sample to decay, to change to nitrogen-14. And that is unaffected by temperature, pressure, or any other variable. We know that the half-life of carbon-14 is 5,730 years. So using those figures, we can translate the loss of carbon-14 in a fossil into an estimate of its absolute time. This is just a picture showing that the 2 atoms of the same element that differ in the number of neutrons is the definition of an isotope. And here they're comparing carbon-12, carbon-13, and carbon-14. And this is again showing the idea that an isotope has a fixed half-life, which again is the number of years it takes for half of it to decay. Another isotope that's present in volcanic rock is used for dating as well, and that's uranium-238. It has a half-life of 4.5 billion years. And so while it's not present in a living organism,